With the enzyme-linked moniker, Dr. Jaffe, will you explain whether enzymes within the lymphocytes are reacting and what is being measured?
That question gets to the heart of what is distinctive and different about ELISA ACT from all other ELISA tests. ELISA ACT is the unique bringing together of enzyme-linked immune-sorbed assay, that is an amplification assay, and ACT, advanced cell culture technique. The difference is profound. The classic ELISA, or ELISA if you prefer, was developed in the 1950s by Burson and Yalow. It was so important in clinical chemistry that they received the Nobel Prize for this technique that involves multiple steps. It involves 5 steps to build up what's called a sandwich. That's just an acronym or a moniker that's given by laboratorians, but 5 steps, each of which adds 3% variance. Trust me, if we go through the intrinsic math, every time you do a step in a lab, you add at least 3%, maybe more, in variance or variability or uncertainty to your results. So the good news about classic ELISA is it is a sandwich that amplifies the presence of small amounts of compounds that can be measured accurately with a high precision antibody. However, at best, 5 steps times 3% is 15% variance. That's the best that a standard sandwich ELISA test tube in vitro test can do. Now let's look in contrast. What did we accomplish at ELISA ACT Biotechnologies to produce the ELISA ACT procedure? Well, first of all, we wanted an ex vivo specimen. We wanted a specimen where we were watching in the laboratory the reaction of living cells just as they react in the body. So that means we had to have people prepare, we had to take the specimen in a very specific way, we had to ship it under very controlled conditions, we had to bring it back up to body temperature in the laboratory, incubate it with these various substances, and then read the result. Now, what's interesting about ELISA ACT is it's a one-step procedure. So you pre-coat the foreign substance on the bottom of the well or the dish. Imagine that there are 48 different wells, each of which is coated with a different substance. And what happens when we put the cell-rich plasma, notice we get the red cells out by gently spinning, and then we have the cell-rich plasma, which contains everything in the plasma, all the cells, including platelets and antigen-presenting cells, APC, antigen-presenting cells, and lymphocytes, T cells, B cells, all kinds of lymphocytes. So we wanted an assay that was ex vivo, we wanted an assay that was functional, we wanted an assay that was high precision, we wanted an assay that used an enzyme in the membrane of the cell that is at the recognition site. In fact, it's involved with the process of turning the cell on when the cell has been programmed against a foreign substance before and it sees it again. Both T cells and B cells activate through antigen. T cells without antibody, B cells through antibody. But in both cases, you have to present the antigen and you have to have an enzyme that turns on only when the cell activates against something foreign. In fact, if you start with our procedure and continue to feed the cells for 3 weeks, you would see a colony of cells form, a proliferation of cells. If you use radioactive isotopes, as was used when we introduced the LRA testing by ELISA ACT method, you would see 3 days to incorporate radioactive thymidine, and now you were down to a mere 15% variance, whereas a one-step amplified procedure, the way we do it in the LRA testing, that one-step procedure has a less than 3% variance, and we put ourselves to the test a few years ago, running 3 consecutive years of split samples with no dropout of data, no outlier data. and less than 3% variance reported at Experimental Biology, American Society for Investigative Pathology, a few years ago. So a novel procedure, the only one-step amplified procedure of which we know, and most importantly, the most precise functional measure, ex vivo, not in vitro, measuring just as it occurs in the body, the reactions in a way that are amplified by an enzyme, a kinase specifically. A kinase adds phosphate to certain molecules and then sends them on a mission. The mission could be to turn on the nucleus so the cells divide. The mission could be activate the cell surface so that the glycocalyx, or what's commonly called the hair on the surface of the cell, activates. And we read that reaction under a microscope. So we know we're looking at lymphocytes. We're not looking at debris. We're not looking at things at a certain size. We're not looking at things other than the way the body looks at them. And just as a footnote, some labs make a point that they test both cooked and raw foods for antibody reaction. Now, there's two problems there. First of all, when you cook a food, you denature it. You make it into random protein or random amino acid groups or globs or messes. But by definition, when you cook the food, you denature the very structure that you want to measure for hypersensitivity. And the other is that if you're measuring antibodies, you're not measuring T cells. You're doing a test tube in vitro test in contrast to the LRA, which is ex vivo. And you have a lower precision because you're measuring those antibodies, whatever the starting material is, you're measuring those antibodies in the classic Burson and Yellow 5-step procedure with at least 15% variance. So you can get functionally more precise results, more reproducible results, more clinically relevant results when you have a one-step procedure and a procedure that is ex vivo, or allowing the body to react just as it does inside, but under the laboratory controlled conditions. So thanks for asking, very important distinction. There are thousands of standard sandwich ELISA procedures. There's only one ELISA Act. There is only one lymphocyte response assay, LRA, done by the ELISA Act procedure. So when you want precision, when you want reproducibility, when you want clinically predictive and actionable information, go ex vivo and go with LRA.